Part 1, A Foundation in Science, London, 1938. Rosalind Franklin stared at the columns of numbers in front of her. Even at 18, her methodical mind refused to gloss over small details. She tapped her pencil lightly on the edge of the desk. Outside, the city was bracing itself for the possibility of war, radio broadcasts crackling with anxious news updates. Inside, however, young Rosalind was in her own world of mathematics, geometry, and chemistry. From a young age, Rosalind's determination and intellect set her apart. She was raised in a respected Anglo-Jewish family that valued education and public service. Her father, Ellis Franklin, was supportive yet traditional. He admired intellect, but there was always an undercurrent. Growing up female in the early 20th century meant navigating certain expectations. Nevertheless, Ellis made sure his daughter had access to the best schooling possible, including St. Paul's Girls' School, renowned for its rigorous academics in the sciences for girls. That afternoon, Rosalind's mother, Muriel, peered into her daughter's room. "Rosie dear, have you forgotten about dinner?" she asked gently, using the family's preferred nickname for her. Startled, Rosalind looked up. "Sorry, Mother. Time got away from me." She closed her notebook with a small sigh. She knew the country teetered on the brink of conflict, but a part of her felt an even stronger tension within. How to keep forging a path in science amid the constant reminders that women didn't always stay the course. Her teachers, seeing her aptitude, strongly encouraged her to pursue higher education in chemistry or physics. Family friends sometimes offered the mild, patronizing question, "You're so clever. Have you considered teaching, my dear?" She wanted research. She wanted discovery. A war-changed world. Rosalind entered Newnham College at Cambridge University in 1938. Shortly afterward, Britain was at war. Her years at Cambridge, shaped by global conflict, would be marked by determination. She immersed herself in chemistry, one of few women in a male-dominated environment. When she wasn't in lectures or labs, she was volunteering in war-related efforts or reading the latest scientific journals. It was at Cambridge that she began to see a bigger picture of how science could profoundly reshape our understanding of life. She worked under some formidable scientists, absorbing rigorous methodology and an uncompromising approach to data, traits that would define her career. Conversations in the lab hallways buzzed about new experiments, better techniques. X-ray crystallography was on the rise, an approach used to deduce the structure of crystals by analyzing how X-rays diffracted off them. While she didn't know it yet, this technique would be the key to her most significant work. Early sparks of DNA research. At that time, DNA, deoxyribonucleic acid, was a curious molecule with an uncertain function. Many scientists believed proteins, not DNA, carried hereditary information. The transforming principle, identified by Frederick Griffith in the late 1920s, was known. But the identity of the hereditary material was still fiercely debated. While Oswald Avery would famously suggest DNA was this genetic substance in 1944, the final proof, especially of its structure, remained elusive. Rosalind was aware of these developments. In a conversation with a fellow student one afternoon in 1940, she flipped through a copy of a journal discussing the Avery-McLeod-McCarty experiment. "I read it last night," she said, with a note of excitement in her voice, pointing to a diagram. "They've shown that DNA can transform bacteria. If this is reproducible and truly the genetic material, imagine what it implies for understanding inheritance, disease, everything." Her companion, a tall young man named Brian, shrugged. "Proteins are more complex. Hard to see a simple chemical like DNA being the blueprint for life." Rosalind's eyes gleamed. "Sometimes simplicity in structure can conceal complexity in function," she said, quoting one of her professors. "If Avery's right, then we're on the cusp of something that could reshape biology." A brief detour, the coal and carbon years. After completing her studies, Rosalind served as an assistant research officer for the British Coal Utilisation Research Association, BCURA. She analysed porosity in coal, eventually leading to influential publications, and a PhD from Cambridge in 1945. Her meticulousness in studying carbon's microstructures and her mastery of X-ray diffraction techniques laid the groundwork for her future. She never viewed her work with carbon as a sideline. It was a rigorous training that honed her eye for detail. She learned to interpret patterns in the X-ray diffraction photos with exacting precision. She came to appreciate how small structural changes at the atomic level could have sweeping implications for material properties. Moving to Paris. One day in late 1946, she received an offer to work in Paris at the Laboratoire Central des Services Chimiques de l'État. There, under the mentorship of crystallographer Jacques Meringue, Franklin refined her mastery of X-ray diffraction. The laboratory atmosphere was different from the sometimes stifling environment she'd experienced in British institutions. In Paris, she found camaraderie, intellectual openness, and a refreshing acceptance of women in science, relatively speaking. Over cups of espresso in small Parisian cafes, she would chat animatedly with colleagues about diffraction patterns, the latest methods in crystallography, and the potential for unveiling the secrets of molecules. She grew into a confident scientist. Outside the lab, she took pleasure in Paris's art and culture, strolling along the Seine, occasionally indulging in French cuisine. But in the lab, she was as driven as ever. A new opportunity, King's College London. In 1950, John Randall, head of the Medical Research Council, MRC, Biophysics Research Unit at King's College London, approached Franklin about joining their team. They had acquired a new X-ray diffraction unit, and they needed a skilled crystallographer to tackle the problem of DNA's structure. Maurice Wilkins was already working there, along with graduate student Raymond Gosling. The letter was both enticing and slightly vague about her exact role. Still, the lure of returning to London, coupled with an opportunity to study DNA with state-of-the-art equipment, was too promising to resist. "Well, Rosie," Jacques Meringue said one morning in the lab, sliding a typed letter across the table. "They want you to be part of a revolution, no?" Rosalind smiled softly. She had grown to admire Meringue's guidance. "I suppose so, but the job description is a bit unclear. Wilkins is apparently the lead researcher. I'm not sure how they want to divide the responsibility." "Be careful, Rosalind," he said, leaning forward. "You have a gift for seeing patterns others cannot. Make sure your contributions are recognized." She nodded, pocketing the letter. "I've become accustomed to forging my own path," she said, with a lightness in her tone that still carried underlying resolve. Thus began the next chapter of her life, one that would pit her in a complex interplay of collaboration and conflict, and lead her to produce some of the most famous photographs in the history of science. Part 2. Arrival at King's College. London, January 1951. Rosalind Franklin stepped onto the bustling Strand and paused in front of the Gothic facade of King's College London. A damp winter chill hung in the air, typical of the city. She clutched her satchel, which contained a notebook of preliminary ideas about DNA's structure. The war was over, but the city still bore its scars. Bomb-damaged buildings, rubble piles in certain districts. Yet there was a hum of renewal and possibility. She made her way to the corridors of the Biophysics Research Unit. The hum of new machinery, the clank of lab apparatus, and the echo of footsteps greeted her. At the end of a hallway, a tall, somewhat reserved-looking man approached. "Dr. Franklin?" he asked, extending his hand. "I'm Maurice Wilkins." "Yes, good to meet you," she replied, shaking his hand firmly. Wilkins's demeanor was polite but cautious. They exchanged a few pleasantries. Wilkins mentioned the lab's latest acquisitions. "We have a fine microcamera for X-ray work, and of course, the MRC funding for exploring the structure of DNA." She was eager, and she assumed they would be working as partners, though she had heard various accounts of Wilkins. Some colleagues hinted he expected a less independent colleague. Randall's letter had implied Rosalind would lead the project. In those first weeks, the environment felt ambiguous. Wilkins seemed to view Rosalind as an assistant. She perceived she was hired as a principal investigator on the DNA project. Neither was entirely wrong. Randall's own communications had sown confusion. The lab and the first clashes. Raymond Gosling, a graduate student, was assigned to assist Rosalind. She immediately took a liking to his earnest attitude. He showed her around the lab's new X-ray apparatus, and they set about refining the technique to obtain the clearest diffraction patterns of DNA samples possible. Let's start with B-form DNA, Rosalind said, setting a slender glass tube on the bench. We'll vary the hydration levels carefully. I suspect water content drastically affects the form it takes. Gosling nodded. Wilkins has been working mostly with A-form. He rarely mentions B-form, something about it being more variable. Rosalind took a breath. We must systematically analyze both forms. DNA is notoriously sensitive to humidity. Their first few attempts were promising. Rosalind's attention to detail in controlling temperature and humidity soon began yielding sharper images. Meanwhile, Wilkins focused on other tasks and would occasionally peek into the lab. One late afternoon, Rosalind was studying a new diffraction pattern, capturing lines and arcs that hinted at a repeating structure. Wilkins entered, nodding at her. "How are the photos looking?" he inquired. "Better than before," she replied, keeping her focus on the developing plate. "We're narrowing the focal distance and controlling conditions more rigorously. There's a large repeat, possibly around 3.4 angstroms." Wilkins peered over her shoulder. "That's the same dimension Watson and Crick over at Cavendish keep talking about," he said, almost offhandedly. "They're working on a model-building approach." Franklin frowned slightly. She'd heard of James Watson and Francis Crick, theoretical types at the Cavendish Laboratory in Cambridge. Rumour had it they were building molecular models without gathering robust experimental data. "They should gather proper evidence," she said, a hint of skepticism in her voice. "Model building is fine, but it must be grounded in solid diffraction results." Wilkins gave a light shrug. "They're quite enthusiastic. They believe the structure might be helical." Rosalind carefully jotted notes in her lab book. "Perhaps. The diffraction pattern suggests some kind of helix, but we need more consistent data. We can't jump to conclusions." Wilkins paused. "Yes, indeed." But something in his tone suggested he was more sympathetic to Watson and Crick's approach than Rosalind was. Across the scientific divide, Cambridge. Meanwhile, at the Cavendish Laboratory, James Watson, a young American postdoctoral researcher, was hunched over a makeshift model of cardboard cutouts and wire rods. Francis Crick, older and gregarious, paced around the table. "Rosalind Franklin's data might be the key," Watson said, adjusting a piece of wire. "Wilkins hinted that she's getting X-ray diffraction pictures of remarkable clarity." Crick let out a short laugh. "Yes, but how do we get her to share them? We haven't exactly established a cordial relationship. From what I hear, she doesn't like our approach." They both knew that in the world of science, data could be gleaned at conferences, through letters or informal discussions. Watson, impulsive and sometimes tactless, would later recall the complicated dance of trying to persuade Wilkins or Franklin to disclose more details. Tensions rise. Back at King's College, misunderstandings between Franklin and Wilkins deepened. The root was partly the lab hierarchy. Wilkins expected a collaborator who would consult him on every major step. Franklin, believing she was independent, pushed forward unilaterally. She was confident in her approach and delivered instructions to Gosling with precision. "Take another set of exposures at 92% relative humidity," she told Gosling. "Let's see if the pattern shifts further. Also, we'll test lower humidity tomorrow for a different form." One day, while examining a fresh negative, Wilkins casually referred to Rosalind as Miss Franklin within her earshot. She stiffened. She had a doctoral degree. The social norms of the time often relegated female scientists to less formal addresses. "Dr. Franklin," she corrected calmly, though her voice was tight. Wilkins frowned, momentarily puzzled. "Yes, of course, Dr. Franklin." But the damage was done. Small slips like that reinforced the wedge between them. Later that evening, Franklin confided in Gosling. "I don't think Wilkins realizes how it sounds, but I need professional respect. We're in a race to uncover the structure of DNA, and I can't afford to be hampered by outdated formalities." Gosling nodded sympathetically. He was stuck in the middle, respecting his supervisor's brilliance, but also knowing Wilkins had authority in the department. A glimmer of breakthrough. Despite interpersonal strains, the scientific progress was accelerating. By mid-1951, Franklin and Gosling had produced diffraction images that hinted strongly at a helical structure, particularly in DNA's B form under higher humidity. She carefully interpreted the patterns, distinct arcs, and a cross-shaped arrangement in the center. The distances between certain arcs suggested a repeating unit of around 3.4 angstroms along the fiber axis, matching the known dimension of bases stacked in DNA. She wasn't yet fully convinced it was a double helix. The data pointed to a helix, but the exact arrangement, 2 strands or 3, oriented parallel or anti-parallel, remained unclear. One afternoon, Wilkins stepped into the darkroom, watching the photo develop under the red safe light. He let out a quiet gasp. "That's quite something," he murmured, seeing the striking cross of spots that indicated a helix. Rosalind suppressed a smile. She was proud of the result. "Yes, we're nearly certain the B form is helical. I'll be refining the molecular parameters next." Wilkins nodded. "We should let Watson and Crick see this. They might—" "No," she cut him off gently but firmly. "Not until I'm fully confident in our analysis. They have a habit of jumping the gun." A flash of irritation crossed Wilkins's face. He'd known Watson and Crick from earlier conferences. He found them brash, but also thought their model-building approach might be complementary. However, Franklin's desire to verify every detail was in direct conflict with Watson, and Crick's eagerness for a bold hypothesis. So the seeds of a competition were sown. On one side, Watson and Crick, constructing hypothetical models, longing for the crucial data that could confirm or refute them. On the other, Franklin, meticulously gathering evidence, determined not to publish or share half-formed conclusions. Social undercurrents. Outside the lab, the culture of King's College didn't help. Women researchers often felt marginalized, with limited access to common rooms and dining halls. Franklin, being both forthright and a woman in a male-dominated environment, found herself socially isolated. In hushed tones, administrators referred to her as difficult or prickly. She didn't bother to soften her directness. Time was too precious, and the research too important. But behind the reserved, sometimes steely facade, Franklin grappled with her own pressures, the precariousness of her role, the overshadowing presence of Wilkins, and the knowledge that Watson and Crick were sprinting forward at Cambridge. She believed wholeheartedly in the power of evidence, certain that if she let the data guide her, truth would emerge clearly. The stage is set. By late 1951, the DNA puzzle began drawing more attention in the scientific community. Publishers of Nature and other journals awaited new revelations. Rumors floated that Linus Pauling, the American chemist who had solved the alpha helix structure of proteins, was turning his formidable mind toward DNA. The race felt global, urgent. Rosalind Franklin's story was already deeply intertwined with that of DNA. Little did she realize how her photos, particularly one labeled Photo 51, would become the definitive clue. Nor could she foresee how precarious her recognition would be in the male-dominated, competitive world of 1950s science. In a quiet moment in the lab after everyone else had gone, Franklin examined the latest set of diffraction images. She ran her fingertips lightly across the pattern. "This is the blueprint of life itself," she thought. "If only we can read it correctly." She turned off the overhead lamp, leaving only the soft glow of the X-ray apparatus indicator. Tomorrow would bring another day of painstaking alignment, calibration, and measurement. Perhaps, somewhere in those patterns of light and shadow, the fundamental secret of living organisms was waiting, soon to be revealed. Part 3. The Race Heats Up. Cambridge, October 1951. James Watson paced the narrow office he shared with Francis Crick, excitement flashing in his bright eyes. A rumor had reached them that Rosalind Franklin's most recent diffraction images were even clearer than before, images that might prove the double helix was correct. Crick, leaning against a cluttered desk, toyed with a plastic model of a single nucleotide. "If we're right about the base pairs forming crosslinks, we need that crucial piece of data on the symmetry and the spacing," he said. "Otherwise, we're just guessing." Watson nodded emphatically. "We need more direct evidence, something we can measure. Wilkins mentioned Rosalind is cautious about sharing. Maybe we can schedule a visit to King's?" Crick stroked his chin. "It's worth a try. We'll be diplomatic. An ill-fated meeting. A few days later, Watson took the train to London, planning to meet Franklin under the pretext of scientific collaboration. He arrived at King's College, making his way through the corridors to the biophysics unit. He poked his head into an office and found Wilkins, half buried in notes. "Ah, Watson," Wilkins said, his tone flat but polite. "You're here to see Rosalind, I gather. She's in the darkroom at the moment." Watson gave a grin that didn't quite mask his impatience. "Yes, I wanted to discuss her recent findings. Perhaps we can compare them with our model building approach." Wilkins stood, exhaling. "I'll introduce you, but, well, just be careful. She doesn't appreciate speculation without thorough data." They reached the darkroom. Wilkins knocked lightly, then opened the door. Inside, Franklin was transferring a wet negative to a drying tray. The soft glow of a red light illuminated her focused expression. "Rosalind," Wilkins said softly, "James Watson from Cambridge is here. He'd like to hear about your results." Franklin turned around, eyebrows arched. She gave Watson a brief, measured look. "Dr. Watson," she acknowledged coolly, "I've heard much about your model building. I hope you've brought some data to compare with ours." Watson cleared his throat. "I'm eager to see your latest photographs and measurements if you're willing to share. We think a double helix is the most likely structure." She crossed her arms, protective of her results. "I am still refining the data, particularly the water content dependency. My findings are not yet conclusive." Watson pressed on. "But if it's helical, as we suspect, you might want to test a two-chain model with the bases on the inside?" Franklin's patience frayed. "Might want to," she repeated icily. "We need conclusive diffraction patterns to confirm that. Conjecture is easy, Dr. Watson, but proof is more demanding." A tense silence fell. Wilkins stood by, feeling the rising friction. Watson, not one for subtlety, tried a different angle. "Well, you do want to solve this before Linus Pauling does, right? He's known to be building models day and night." Franklin bristled. "Science isn't a horse race to me. If your model can't stand up to actual data, it's worthless. I'm not here to indulge guesswork. Good afternoon, Dr. Watson." The dismissal was clear. Watson, red-eared, muttered an awkward farewell and retreated. Wilkins lingered for a moment, then slipped out. Alone again, Franklin let out a long breath. The mention of Pauling only added to the sense of competition swirling around them. She returned to the negative, focusing on the measured arcs, the distances. Truth would emerge from the meticulous analysis of these images, she believed, yet she sensed the pressure intensifying. The model that failed. Back in Cambridge, Watson, incensed by the encounter, was more determined than ever to prove the double helix structure. He and Crick embarked on a rapid attempt to assemble a model for DNA. They placed sugar phosphate backbones on the inside, bases outward. When they proudly showed it to a visiting Rosalind Franklin a few weeks later, arranged by a reluctant Wilkins, she quickly spotted a fatal flaw. The phosphate groups, with their negative charges being on the inside, made little chemical sense in an aqueous environment. "This is nonsense," she said bluntly. "The molecule would fly apart electrostatically." Watson and Crick flushed. Their immediate attempt at DNA structure collapsed, resulting in an embarrassing retreat when Sir Lawrence Bragg, head of the Cavendish laboratory, told them to drop DNA research for a while. Rosalind's calculations grow stronger. Meanwhile, Franklin, bolstered by Gosling's diligent work, refined her measurements. By early 1952, they had perfected a technique that yielded the legendary Photo 51 of the B form of DNA. This image was exquisitely sharp. A bold X pattern that strongly indicated a helical structure with specific repeating measurements along the axis. She methodically analyzed the diffraction spots, calculating the helical parameters. The angle of the cross suggested the pitch of the helix. The layering lines indicated the distance between stacked bases, approximately 3-4 angstroms. The heavy arcs gave clues to the overall diameter. She was on the verge of writing up her findings in a comprehensive manner, possibly to submit to nature. Still, friction in the lab intensified. Wilkins felt sidelined from the big breakthrough. Franklin felt that her independence was under constant threat, Occasionally, she locked away her notebooks, worried about data leaking prematurely. Broader scientific context, 1952. Beyond the King's College conflicts, the scientific world was abuzz with new revelations. Erwin Chargaff had shown that in DNA, adenine, A, matches thymine, T, in proportion, and cytosine, C, matches guanine, G. This ratio strongly hinted at complementary base pairing. Linus Pauling published a partial guess for DNA, but it contained major errors, a triple helix with phosphate groups inside. Maurice Wilkins presented some of King's data at conferences, enough to spark curiosity, but not enough to overshadow Franklin. Late that year, while Franklin was away at a conference, Wilkins, possibly out of frustration or seeking a second opinion, showed photo 51 to James Watson when he visited King's. This moment, a pivotal historical footnote, would become a point of deep contention. Franklin never explicitly authorized sharing that particular image. Watson's eyes widened the instant he saw it. In his later recollections, he described having an epiphany that DNA must be a double helix. Yet at the time, he did not fully grasp the exact parameters. He just knew it was the missing puzzle piece he and Crick had needed. A chance encounter. On a cold afternoon in December 1952, Rosalind left King's, stepping onto the busy street. She was heading to lunch when quite unexpectedly she bumped into Francis Crick, who was visiting London for a separate meeting. "Dr. Franklin," Crick said, surprised but cordial. "I was just on my way to a talk. How is the work?" Her eyes flicked over him warily. She had a better opinion of Crick than of Watson. Crick seemed more measured, more respectful of data. "It progresses," she replied. "I'm analyzing the finer details of the B-form. The structure is almost certainly helical." Crick gave a faint smile. "We have our own ideas about base pairing, but we're missing precise measurements, particularly the diameter of the helix and the vertical rise per nucleotide." "Data matters," she said simply. "Without it, any model is speculation." He nodded. "Indeed. But speculation can drive the search for the right data, yes?" She pursed her lips, neither agreeing nor disagreeing. "Good day, Dr. Crick. I'm sure we'll all see each other's results soon enough." They parted ways, and Rosalind couldn't help wondering if the competitive fervor overshadowed the excitement of pure discovery. She wanted the truth to emerge clearly, based on unquestionable evidence, health concerns and shifting focus. Around this time, Franklin began suffering from occasional abdominal pains. She didn't think much of it. Long hours hunched over a diffraction camera or analyzing images left her exhausted. She brushed it off, attributing it to stress. Her relationship with Wilkins had deteriorated to the point that John Randall intervened, suggesting that Franklin might move to Birkbeck College to work on the structure of viruses after she finished her DNA analysis. She began to consider the transition. While she felt a sting, was she being pushed out? She also looked forward to leading her own group without the daily tensions at King's. Culmination of the race. By early 1953, a series of small events led to a major leap in understanding. Watson sees photo 51. Though the exact date is debated, by January 1953, James Watson had viewed the photograph Wilkins shared. Crick gains Franklin's data. A lab colleague, Max Perutz, had an MRC report containing some of Franklin's numbers on the unit cell dimensions and water content. It somehow found its way into Crick's hands. While not explicitly confidential, Franklin hadn't planned for it to circulate widely. Chargaff's ratios. Meanwhile, Watson and Crick fully embraced Erwin Chargaff's findings that A equals sign T and C equals sign G. With these crucial pieces, Watson and Crick frantically resumed model building. They placed the sugar-phosphate backbones on the outside, the bases pointing inward, pairing A with T, C with G. Each base pair matched the known helical dimensions gleaned from Franklin's data. As they fine-tuned the model, it seemed to click into place. The double helix explained Chargaff's ratios, the X-ray diffraction data, and the chemical stability. Rosalind's quiet realization. Rosalind, nearing her departure from King's to Birkbeck, was finalizing her own manuscript on DNA's structure. She had all but concluded it was a double helix with anti-parallel strands. She was days or weeks away from detailing the arrangement of phosphates and bases. One evening she stood in front of her pinned-up diffraction patterns. She mentally overlaid them with the possible molecular arrangement. The logic was inescapable. If the bases stacked at 3.4 angstrom intervals and each turn of the helix had about 10 residues that put the pitch near 34 angstroms, exactly what her data suggested. Despite feeling the joy of nearing a significant discovery, she also sensed the storm outside her lab door. Watson and Crick, Wilkins, Pauling. Everyone was converging on the same conclusion. She only hoped that her careful, data-driven approach would earn her the recognition she deserved, the ticking clock. By March 1953, the pieces were moving quickly. Crick was confident enough to proclaim, somewhat boastfully, that they had found the secret of life. Watson hammered out a draft of a paper for Nature, which included the now famous line about the complementary base pairs, pointing out a possible copying mechanism for the genetic material. Rosalind sensed the tension crackling in the air. She saw an early draft of Watson and Crick's structure, or at least heard a detailed description from Wilkins. It infuriated her that they had used her data, some of it gleaned without her direct permission, but she could not deny the correctness of the model. In a twist of fate, John Randall and others decided that the final publication in Nature would feature 3 back-to-back articles, one by Watson and Crick presenting the model, one by Wilkins and colleagues, and one by Franklin and Gosling. It was a way to acknowledge the contributions from both King's College and Cambridge, albeit an arrangement that many saw as overshadowing Franklin's singular role. In these last days before the papers went to press, the stage was set for the unveiling of DNA's structure, a scientific milestone that would forever change biology, medicine, and our understanding of life itself. Part 4. The Unveiling and the Aftermath. Cambridge, March 1953. Francis Crick read over the final manuscript that he and Watson were about to submit to Nature. It was a short piece, astonishingly brief for such a monumental claim. The essential statement was that DNA existed as 2 helical chains, each coiled around the same axis, and crucially the chains were held together by hydrogen bonds between pairs of bases, adenine with thymine and guanine with cytosine. Across the table, Watson fiddled with a pen. "Should we explicitly mention Franklin's photograph or data?" he asked, a hint of anxiety in his voice. Crick tapped the page. "We give credit to Dr. Wilkins for providing us with information about the X-ray diffraction results, but since Franklin and Gosling's paper will be in the same issue, I think that covers it." Watson wasn't entirely comfortable, but he also recognized that acknowledging Franklin's contribution too explicitly might highlight how central her data had been. They wanted the focus on the double helical model itself, King's College. At King's, Rosalind Franklin and Raymond Gosling were putting the finishing touches on their own manuscript. Franklin's paper detailed the B form of DNA, describing the precise parameters gleaned from X-ray diffraction. It was pure experimental rigor. She clarified the methodology, how they managed humidity control, how they measured the reflection angles, and how they concluded a helical structure. Maurice Wilkins, with 2 colleagues, was also finalizing a 3rd paper describing additional supporting data from King's. The plan. All 3 would appear in Nature back-to-back, with Watson and Crick's model in the lead. The publication. On April 25, 1953, the 3 papers were published in Nature. Watson and Crick's paper famously ended with the understated sentence, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." It was a bombshell in the scientific community. Rosalind Franklin's paper meticulously presented the evidence for a helical structure of the B form. Wilkins's group provided further confirmation. In effect, the synergy of the 3 contributions collectively solved DNA's structure. But in the public eye, Watson and Crick's succinct and bold claim seized the spotlight. Reactions and private feelings. At a King's College cafeteria, a small radio set crackled with news about the new discovery in the chemistry of heredity. Rosalind sat with Gosling, sipping tea. A couple of staff members congratulated her, but the grandeur of the discovery overshadowed any individual praise. "How do you feel about it all?" Gosling ventured, his voice quiet. She sighed. "Pleased that the structure is out there. Disappointed by the credit distribution." She set down her teacup. Still, the data is published. That's what matters. Gosling nodded. He admired her dedication, but he saw the injustice. Without Photo 51 and Franklin's analyses, Watson and Crick might not have been able to confirm their model so decisively. A final exchange. Maurice Wilkins approached Rosalind that afternoon. "Congratulations, Rosalind. The pictures and your analysis were crucial." He paused, fiddling with a pen. "Perhaps we should, well, bury the hatchet." She studied him for a moment, her expression unreadable. "You could have come to me directly about sharing Photo 51 with Watson," she said. "I would have given you a version once the analysis was complete." He looked away. Yes, I see now, but the important thing is that the truth is known." Rosalind exhaled slowly. "Yes, the truth is known." They parted with a subdued handshake. Neither was fully satisfied, but they recognized the magnitude of the discovery overshadowed personal tensions. Transition to Birkbeck. Shortly after the publication, Franklin officially moved to Birkbeck College under John Burnell, a renowned crystallographer. She shifted her focus to studying the structure of tobacco mosaic virus, TMV, and later the polio virus. Her skill in X-ray diffraction again shone, producing groundbreaking insights. In her new environment, she found more respect and independence. She formed a small, vibrant research group, including future Nobel Prize winner Aaron Klug. The atmosphere at Birkbeck contrasted sharply with King's. She was happier, freer, though the demands of her work were no less intense. A race won, a legacy uncertain. Meanwhile, Watson and Crick basked in growing fame. They gave talks at universities and scientific conferences, their double helix model hailed as a revolutionary breakthrough. Newspapers ran headlines about the secret of life being uncovered in Cambridge. Rosalind Franklin's name was mentioned in scientific circles, but in the public sphere, her contribution was often overshadowed. For her part, she continued her virus studies with characteristic rigor, publishing a series of papers that laid foundations for structural virology. Illness and determination. By 1956, Franklin's health began to deteriorate. She was diagnosed with ovarian cancer. Despite surgeries and treatments, she continued working, sometimes returning to the lab shortly after hospitalization. Friends noticed her becoming more reflective but no less dedicated. Aaron Klug later remarked how stoically she handled the illness. She rarely complained, pouring her energies into finishing as much work as possible. In a quiet conversation with Klug in the Birkbeck lab one evening, she said, "Whatever I've accomplished is only meaningful if it helps the next generation push further. Whether in DNA or viruses, the structure is just the beginning. We must apply what we learn to understand life and cure diseases, I hope." She gave a wan smile, pressing a hand to her side where the pain flared. Recognitions and oversights. During these years, Watson, Crick, and Wilkins continued to refine the DNA model. In 1962, the Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins for their discovery of the structure of DNA. Rosalind Franklin had passed away 4 years earlier, on April 16, 1958, at the age of 37. Nobel Prizes are not awarded posthumously, so she would not have been eligible even had the committee considered her. In the wake of that Nobel announcement, some in the scientific community began to revisit Franklin's contributions. Would things have been different if she had lived longer? Could she have shared in that pinnacle of recognition a lasting impression? At Birkbeck, her colleagues mourned her passing. In the lab's quiet corners, pinned photographs and diffraction patterns told the story of a brilliant crystallographer whose unwavering commitment to empirical evidence had helped reveal the fundamental secret of how life stores and transmits information. In later years, as Watson wrote books and gave interviews, as Crick moved on to neuroscience, the story of Rosalind Franklin's Photo 51 and her role in understanding DNA slowly came to the public's attention. Historians of science and feminist scholars championed her achievements, citing the structural sexism of mid-century academia that impeded her rightful status as a co-discoverer. Yet in her time, Rosalind herself remained primarily focused on the science rather than the accolades. As she once wrote to a friend, "Science and everyday life cannot and should not be separated. Science, for me, gives a partial explanation of life. Insofar as it goes, it is based on fact, experience, and experiment. Epilogue. Today, the double helix structure of DNA is central to everything from genetics and molecular biology to forensic science and evolutionary studies. The nascent field of genomics has become a driving force in modern medicine, leading to insights into heredity, disease mechanisms, and personalized treatments. The Human Genome Project, CRISPR gene editing, and countless breakthroughs all trace back to that pivotal moment when X-ray images, careful measurements, and a bit of daring model building converged to reveal life's blueprint. And behind that triumph was Rosalind Franklin, resolute, brilliant, sometimes uncompromising, always devoted to the truth. Though overshadowed by her male counterparts at the time, her legacy shines ever brighter with each passing year. Her photographs, especially Photo 51, remain an iconic testament to the power of careful, meticulous science. Whenever a student of biology opens a textbook to the page describing the double helix, Rosalind Franklin's spirit lingers in that classic X-shaped diffraction pattern. She was the one who captured the fleeting shadows of life's molecular spiral, illuminating a path for generations to come. Part 5. The Science of DNA and Franklin's Legacy. Note: this segment delves more deeply into the scientific background of DNA, elaborates on Rosalind Franklin's methodology, and discusses the future implications of her work. It also adds narrative elements to maintain story flow. A closer look at DNA. DNA, deoxyribonucleic acid, consists of a sugar phosphate backbone composed of alternating deoxyribose sugar and phosphate groups, 4 nitrogenous bases, adenine, A, thymine, T, cytosine, C, and guanine, G. These bases attach to the sugar, forming nucleotides. Early researchers struggled to see how these components fit together to store genetic information. Proteins, with their 20 amino acids, seemed more plausible as carriers of genetic complexity. DNA, with only 4 repeating bases, seemed too simple, until the pattern of A pairing with T and C pairing with G, Chargaff's rules, emerged, suggesting a pairing mechanism. Rosalind Franklin's method, X-ray diffraction. X-ray diffraction involves directing a beam of X-rays at a crystallized or fibrous sample of a molecule. The X-rays scatter, diffract, upon hitting the electrons of the atoms in the molecule, creating a pattern of spots on photographic film. By measuring the intensities and angles of these spots, scientists can infer the 3-dimensional arrangement of atoms. Franklin's expertise lay in her meticulous sample preparation and her sharp, analytical eye. Sample preparation. DNA fibers are sensitive to humidity. Franklin developed careful protocols to hydrate or dehydrate the fibers to obtain the A form, low hydration, and the B form, high hydration. Alignment. For the diffraction patterns to be interpretable, fibers must be uniformly aligned. Franklin spent hours perfecting techniques to mount DNA fibers so they produced clean, ordered patterns. Measurement. Interpreting the arcs, rings, and crosses in the diffraction pattern required advanced mathematical analysis, determining the distances between atomic planes. Franklin's background in crystallography, from her coal and carbon research, honed her skills for accurately extracting parameters like helical pitch and diameter. What Photo 51 revealed. Photo 51, captured in May 1952, showed a clear cross pattern of spots, typical of a helical structure. The black arcs indicated repeating units along the fiber axis. Notably, the distance between repeating layers, approximately 3, 4A, matched the stacking of nucleotide bases. The diameter implied there were likely 2 strands, given standard atomic radii and the known density of DNA. The symmetry of the pattern suggested the strands were anti-parallel, though Franklin herself was cautious about concluding the arrangement of bases just from one photo. Franklin drafted a summary of these findings, hypothesizing a 2-chain structure. While she did not conclusively announce a double helix in her notes, she provided enough detail that anyone with additional pieces, like Chargaff's ratios or the notion of base complementarity, could piece it together, the intersection of competition and collaboration. Science is often collaborative, but the DNA story highlights the competitive streak that can arise when major discoveries are at stake. Wilkins hoped Franklin would corroborate his data and work in tandem. Their conflicting personalities and confusing directives from Randall led to friction. Watson and Crick needed Franklin's precise data to test their models. Without it, they were stumbling in the dark. Their brilliance in model building meshed perfectly with Franklin's expertise in experimental crystallography. Had the relationship been more cooperative, she might have been recognized on par with them from the outset. Pauling loomed in the background. His groundbreaking work on protein structures made him a likely competitor to solve DNA as well. Indeed, he published a flawed triple helix model, indicating that even a great mind could err without complete data. In a sense, the DNA discovery was an orchestra of different skills, with Franklin's X-ray data forming the symphonic centerpiece. Watson and Crick might have been the conductors who assembled the final performance, but Franklin's notes gave it resonance and clarity. Franklin's later years at Birkbeck. After she left King's in mid-1953, Franklin pivoted to virology. She investigated the rod-like structure of the tobacco mosaic virus, TMV, demonstrating that the viral RNA was embedded in the protein coat, a revelation in understanding viral assembly. She applied similar X-ray diffraction methods to other viruses, collaborating with Aaron Klug. Their work laid the foundation for much of modern structural virology, influencing how scientists approach vaccines and antiviral drugs. Despite her cancer diagnosis, Franklin remained active until the final months of her life. She published numerous papers, mentored younger scientists, and formed friendships built on mutual respect, particularly at Birkbeck, which was more welcoming than King's. The broader impact of DNA's discovery. The identification of DNA's double helix was just the first step. In the decades following 1953, molecular biology exploded. Genetic code. Researchers deciphered how sequences of bases encoded amino acids, revealing the universal genetic code. Recombinant DNA technology. The 1970s and 1980s saw the development of gene cloning and genetic engineering, leading to breakthroughs in medicine, agriculture, and biotechnology. Human Genome Project. Completed in 2003, the project mapped the entire human DNA sequence, revolutionizing our understanding of genes linked to diseases. CRISPR-Cas. 9. Modern gene editing techniques allow precise manipulation of the genome, opening possibilities for curing genetic disorders, but also sparking ethical debates. Franklin did not live to see these extraordinary transformations, but her foundational work on DNA structure was essential, reflecting on gender and recognition in science. Rosalind Franklin's story is emblematic of the challenges faced by women in science during the mid-20th century. She contended with a research environment that often saw women as subordinate. She navigated a labyrinth of social norms, where directness from a woman was interpreted as abrasiveness. Despite her pivotal contributions, full recognition only gradually emerged, in part through the writings of feminist scholars and historians of science. Today, numerous institutions honor her memory. Buildings, scholarships, and scientific awards bear her name, signifying a shift in acknowledging the importance of diversity and fairness in scientific endeavors. A dramatized vignette, Franklin's Contemplation. Below is a short fictionalized scene that illustrates how Franklin might have reflected on her legacy, even if not precisely recorded in history. Birkbeck College, 1957. The evening shadows stretched across the lab benches, where trays of photographic plates lay. Rosalind Franklin, weakened from recent treatments, remained at her workstation. The smell of developing fluid lingered in the air. Franklin, murmuring to herself, DNA, viruses, so much more to uncover. We're barely scratching the surface. The structure is only the beginning. There's replication, transcription, translation. She gently lifted a negative plate against the overhead lamp, squinting at a faint ring pattern. Dr. Aaron Klug, staying late himself, approached. Klug. You should rest, Rosalind. Franklin, with a small smile. I'll rest soon. Just one more look. These capsid proteins, they form such elegant symmetry. She set the plate aside, turning off the light box. Klug. You've achieved extraordinary things. Someday, the world will understand just how vital your work was. Franklin, softly. Someday. But it doesn't matter who gets the credit, does it? As long as the truth is known. They switched off the lab lights, stepping out into the corridor. Although Franklin couldn't foresee her posthumous legacy, she carried the satisfaction of a scientist who believed firmly in the pursuit of knowledge for its own sake. A legacy coming to light. Long after Franklin's passing, biographers such as Anne Sayre and Brenda Maddox wrote extensively about her life, presenting a balanced narrative against James Watson's portrayal in the Double Helix, which, though engaging, had been criticized for dismissive and sometimes unfair depictions of Franklin. Watson himself later acknowledged that some of his descriptions were flippant or insensitive. Over time, the scientific community openly recognized that Franklin's X-ray diffraction results were critical to unraveling DNA's structure. Modern view. Co-discoverer. Many scientists and historians today refer to Rosalind Franklin as a co-discoverer of the DNA double helix, alongside Watson, Crick, and Wilkins. Although she didn't share in the 1962 Nobel Prize, her reputation as one of the 20th century's most important scientists is firmly established. Indeed, had circumstances been different, had she lived longer, or had the lab politics been more transparent, her role might have been acknowledged from the outset. The future she helped shape. From the vantage point of the 21st century, we see how the double helix model paved the way for molecular diagnostics. Genetic tests can identify hereditary predispositions to diseases, guiding prevention or early intervention strategies. Gene therapy. Scientists experiment with ways to replace or fix faulty genes to treat genetic disorders. Personalized medicine. By analyzing individual genetic profiles, doctors can tailor treatments for maximum efficacy and minimal side effects. Evolutionary biology. Comparing DNA sequences across species illuminates the tree of life and unravels the evolutionary relationships among organisms. All these advances trace back to that crucial time in the early 1950s. While Watson and Crick's model was the conceptual leap, Rosalind Franklin's data made the leap possible. It's a testament to how science thrives on the convergence of different talents, methods, and viewpoints, even amid human conflicts and frailties. A final word. Rosalind Franklin's story underscores a timeless lesson. Groundbreaking discoveries rarely emerge from a single source. Rather, they materialize when meticulous data, bold hypothesizing, technical skill, and sheer perseverance intersect. Franklin's unwavering commitment to empirical rigor, her technical brilliance in X-ray crystallography, and her resilience in the face of institutional barriers ensured that her name would forever be linked with one of humanity's greatest scientific achievements. Though her life was cut short, her impact resonates in every genetic test, every virus structure solved, and every genomic breakthrough that builds upon the double helix. When we marvel at the elegance of DNA, we pay silent homage to the extraordinary scientist who helped reveal its spiral secrets.